Welcome to the first technical session, and this is the main reason everybody comes to this meeting, I think. And this morning we have a keynote presentation by Andy Newhouse, who, as you probably all know, Andy's the main reason that we're here. And Andy's been working on the transgenic chestnut now for a long time. Actually, SUNY ESF has been working for a much longer time on this than Andy has. And so all of us are interested to listen to the update, maybe correct some misinformation that has been out there, and now we can get the record set straight. And so I don't want to take any more time from Andy. Take it away.
Thank you for the introduction. Well, I will say again, welcome to Syracuse. We're excited that you're here. I will also, in addition to introducing myself, I have a couple pictures, but I will say right up front that the work and the research that I'm describing is on behalf of a large and very active research group, several of whom are here today. So I'm going to ask, I'm going to put you on the spot, if you're part of our ESF team, can you stand up and wave? Eric, I know there are a couple more in the room. Linda, Hannah over on that side. So you will meet several more of our team members through various talks and tours and presentations throughout these next few days. So please feel free to talk to them. In many cases, they know more about these details than I do, so I might even tell you to talk to them if there are questions that I personally can't answer. But we're really looking forward to hosting you here and showing you some of what we're working on. This is my family. My name is Andy Newhouse. This is, I have two kids. They are twin boys turning 12 in a few weeks. I also have a new puppy. So we're still getting up in the middle of the night and so on. Right. Well, they're middle school now. It doesn't count. So before I get started with the more technical portion of the talk, I want to remember a couple of people who are really foundational who were the foundation of our research program. So on the left is Bill Powell, and second from right is Chuck Maynard. They founded the research project, the whole idea of the project here at ESF, starting in 1989. And I came here in 2003, I've been working on it ever since. Linda's in this picture also. She's been working on this even longer than I have. So Bill and Chuck both passed away recently. Bill at the end of last year and Chuck at the beginning of this year. And so we're really excited to carry on their legacy, but also really feeling this loss still. Bill and Chuck were both honored from the New York State Senate with these commendation awards at the end of last year. So we're really proud to recognize their contributions. And even more than that, to carry on their legacy, to keep continuing this project that they started. As a brief overview of my talk, I'm going to give a little bit of American Chestnut background. I know this is very familiar to many of you, but I'm going to start with that context, go over our program in a little bit more detail. Last year in Missouri, I gave kind of the super quick overview, and so this year I'm going to go into a little bit more depth. Also, since last year, there have been some updates, some changes, and so I'm going to go over the current status of the darling chestnuts and talk about what we are going to be doing next, what this work is leading into. American chestnuts were, as you are aware, really foundational to much of the eastern U.S. history and culture. Things like street names and Christmas songs reflect that importance. The wood was valuable for a number of purposes, especially outdoor, things like cabins and fences and so on. It was rot resistant. The chestnuts, as many of you know better than I do, are useful for a variety of foods. Can be used for things like baking, and I also brewed a chestnut beer several years ago with help from some of our team members. So lots of benefits to the chestnuts. I'll say right at this point that our intention for working with American chestnuts is not to create a commercial food product. Many of the hybrids are better for many aspects of that, but there are these unique benefits of a forest-type tree that we really rarely see in forests anymore. So. Again, our main intention with the restoration project for American chestnuts is for the environmental benefits, is for the wildlife, is for the forest health, is for the biodiversity. So this is just, these are just examples of some of the types of animals that interact with American chestnut trees, including several species of moths, which were recorded to have gone extinct with the decline of the American chestnut. And a few of these have actually been rediscovered as people are growing chestnuts again. So some of these moths and rare chestnut bee was rediscovered in New York and documented for the first time recently. So we're excited to hopefully see some of those environmental relationships coming back along with the trees. American chestnut was native to, centered around the Appalachian Mountains, much of the eastern U.S., except for the southeastern coasts. Take any range maps with a grain of salt. This is the one that gets used most often from someone named Little in 1977, but there are many different range maps, and even 1977 was well after the decline of the American chestnut, so especially the edges of this range are pretty fluid or are based on guesses from decades ago. So this, where we are now in Syracuse, is kind of the northern part of the range, but these borders are not well defined. That changed, of course, with the introduction of chestnut blight. 1904 is the date that gets used. That's when the fungal organism was identified, or when it was published. It was probably introduced in the late 1800s, and what happens is the fungus is introduced to the trees through a wound, through a crack, through the base of a twig, something like that. And then it secretes several compounds that kill the tissue under the bark. And as that continues, that infection, it's called canker, wraps around the tree, and then the water and nutrients can no longer flow through that part of the stem, and everything above that is going to die. Eventually, the spores will wash down the stem. Once there's a canker at the base of the stem, that whole above ground portion of the tree is going to die. So you can see a historical photo of a mostly infected tree here, but the blight doesn't kill the roots of the tree. And so this is why chestnuts are not extinct or even listed as endangered by most lists, because they're really good at stump sprouting. And so they send up these sprouts if the roots are healthy and well established. And the sprouts can grow really fast. This is 7 feet or so of growth in one season from some re-sprouts. They can grow for several years like this. They're kind of persisting as these shrubby, multi-stemmed bushes, but rarely growing to maturity. They can sometimes flower, but they're clearly not contributing to the ecosystem the same way that chestnuts once did. So we say they're functionally extinct because they're not functioning in the ecosystem in the same way. There are several options for people who want to plant chestnuts, of course, in the wake of chestnut blight. So one of the first ones and one of the first things that was tried even back to the 1930s was planting other species. The blight fungus came from Asia, so if you plant Asian chestnuts that co-evolved with the blight fungus, they tend to tolerate blight a lot better. I put up a picture from Dunstan chestnuts here, not to suggest that that's a particularly good variety, but just that they're widely available. People can buy chestnuts at Walmart, and then they can plant chestnuts. But those trees, Dunstan and many others, are either mostly Chinese or hybrid trees that don't have the same traits as American chestnut. And so I have some contrasts here. The size of the nut itself is very different. In some cases where you want to harvest chestnuts, or if you are the one peeling chestnuts, those big hybrid or Chinese or European chestnuts are clearly favorable. If you're a blue jay trying to eat something, the little American chestnuts are a potential food source, and the golf ball size hybrids are not. So there are clearly wildlife interactions that are going to be different between American and Chinese, as well as the overall tree form, the ability for it to survive and thrive in eastern U.S. forests. Some insect interactions will be different. If you've seen leaves of Chinese and American chestnut, the leaf texture and thickness is very different, and so you can imagine insects are going to interact with those differently, find one easier to chew or more favorable for other reasons. So there are a number of things that can happen when you try to hybridize American and Chinese or other chestnuts, and I put risks in quotes here because these are not things that are dangerous to people. One of the most common is male sterility with certain types of crosses, where if you cross an American chestnut mother tree with Chinese chestnut pollen, some of the offspring don't make viable pollen. But if you continue to breed those, if you put viable pollen onto them, the next generations are generally fine. This is something that's kind of well understood, and just most of these things, like I say in the bottom, can be managed or prevented. They're not considered scary, they're not dangerous to people, it's just sometimes weird stuff happens with hybrid crosses. I've heard about whole generations of crosses between certain trees that just don't germinate. Well, that happens sometimes, and well, don't try that cross again and move on and try a different cross, and it's something that can be managed. So, risks in quotes for some types of hybrids and other types of trees. And I'll also mention that there's kind of a perception within parts of the American chestnut restoration community that Chinese trees are somehow dangerous, that they're somehow bad for restoration or bad for the forests. And that probably came from the idea that the Chinese trees don't have the same traits that allowed American chestnuts to survive and thrive in forests. But that doesn't mean they're dangerous. That doesn't mean they're scary. If they're not going to survive and thrive in our forests, then, well, that's kind of it, and they won't matter in our forests. So, right, I'm going to get into back crossing in a second, but another attempt that was made to enhance blight resistance was called mutagenesis, or exposing seeds to radiation to hopefully induce random mutations. And if you do enough of these, you might get a random mutation that has a trait you like blight resistance was the hope. Tens of thousands of seeds were irradiated throughout a few decades, kind of post World War II, with sometimes extremely high doses of radiation, far higher than what would kill a human. And at the highest doses, most of the seeds were killed too. At the lower doses, did this work? Did this induce blight tolerance? Basically, no. The only published outcome that I could find were people said the trees do not look like other American chestnuts. That was from one visit to a few trees. We have some of these seedlings, like descendants from this mutagenesis program. We have some of these on our research plots, and I think they do look exactly like other American chestnuts. I can't tell them apart. They're American chestnuts with some number of random mutations that didn't make a noticeable difference, benefits or otherwise. So another option for treating or managing chestnut blight is various types of biocontrols. I start with hypovirulence, and Sandy can tell you a lot more than I can about this, but basically the idea is the fungus can get infected by a virus. The virus weakens the fungus, thereby favoring the tree. This can be effective in protecting individual trees, or some cases stands of trees, but the virus doesn't easily spread at a landscape scale. And so this isn't really a viable option to protect or restore trees ecosystem-wide. There are other options like some types of microbes that inhibit the growth of the chestnut blight fungus. And one that you will get to see in person on our field tours on Wednesday is something called mud packing, where you can take soil and basically squish it onto a canker and wrap it in plastic and leave it for a couple of months, and that can heal the canker. It sounds really crude, and it is, but it kind of works. It's, at least most of the time, it can take care of that canker. This doesn't protect the rest of the tree. So if there are several cankers on the tree, it's not going to be a magic cure. But if there's a single kind of discrete canker and you can reach it and you can wrap it up, that can prolong the life of a tree for at least several years. You'll see some examples outside. The other option that's been used most recently is called back-cross breeding. And the theory here is that the Chinese chestnut trees directly are not very well adapted to our forests. American chestnut trees are very susceptible to blight, but otherwise they have all the traits that people want for restoration. And so can you combine those traits and get the best of both worlds? This program has been managed by the American Chestnut Foundation for about the last 40 years. So the idea is, right, combine the American and Chinese chestnut trees. The first generation hybrids have a mix of both of those species, mix of both parents. But then cross that back to American chestnut and hopefully accumulate more of the traits of the American chestnut while retaining the blight tolerance of the Chinese chestnut. And right, goal is 15/16 or 90+% American by inheritance. But this whole thing is based on the assumption that there are just 2 or 3 genes in the genome that control resistance to chestnut blight. It turns out, as we didn't know in the 80s, but we do know now, it's a lot more complicated than that. There are probably a dozen or more significant genes that contribute to blight resistance in Chinese chestnut. And so getting all of those into these offspring without a whole bunch of other Chinese chestnut traits has been very difficult. This program continues. It's being improved through the use of modern technology, but it has not so far resulted in a population of trees with mostly American traits and solid blight resistance. So this is another option, though, for approaches toward enhancing blight resistance. Our approach is more precise. We have one gene or one trait that we're adding into American chestnut trees through genetic engineering. The gene that we are using came from wheat. It's called oxalate oxidase. And if you remember from the blight slide, the way the blight fungus infects the tree, it doesn't eat the living portions of the tree directly. The blight fungus doesn't rely on living tissue. The blight fungus secretes a few compounds that kill the live tissue, and then the fungal organism eats the dead part. This is kind of more understandable understanding the lifestyle of the fungus in China, in Asia, where it's native, where it's called a saprophyte, and it can live on living tissue, but it's more active on the dead tissue. It survives and reproduces on dead chestnut tissue. So oxalate oxidase, this gene that we've added, makes an enzyme that breaks down one of those toxins. It breaks down oxalic acid. And so this mechanism is really unique because it's not a fungicide. It's not killing the fungus. It's definitely not preventing the fungus from infecting the tree. As you'll see, we still see cankers on these trees. The fungus is there. They tend to cause less damage on the tree when oxalate oxidase is present, when the tree can break down one of these toxins the fungus is releasing. So I'm going to use several terms. GMO is probably the most familiar for genetically modified organism. We tend to use genetic engineering or genetically engineered, but transgenic and transformed are all used basically synonymously for this purpose. This sounds tricky. How do we do this? How do we just put a new gene into a tree? You will hear a little bit more about this if you can go to the lab tour on Wednesday morning, but I'm going to try and give an overview here. We use a natural process called agrobacterium transformation. Agrobacterium is a microbe, it's a bacteria that lives in the soil, and its lifestyle involves putting some of its DNA into a tree root. So agrobacterium, this bacteria in the soil, naturally moves bacterial DNA into plants. And in the 1970s, some smart scientists figure out that it always puts in the same little section of DNA into the plant cells. And if we work with it in the lab, we can take out that DNA that it puts into the plant and swap in any DNA we want. almost. So we put the oxalate oxidase gene into Agrobacterium. Oh, I have animations here. So Agrobacterium puts some of its DNA into plant cells. We put in a different gene, a different sequence into the Agrobacterium, which then moves it into the plant cells. But it only does this one cell at a time. So if we put some Agrobacterium on a tree, a few of the cells might take up that new DNA, but it wouldn't protect the rest of the tree. It wouldn't get into every cell in the tree. So we have to start with what are called embryos, which is basically the part inside the seed that will develop into the new tree. So we have some mature chestnuts here. Shows an immature seed, something like you'd see right now, or maybe in a couple weeks, in the summer before it develops all the way. And this is a cross section, so we've cut it in half, and you can see these little blobs at the end. Those are embryos, and that's what's going to start germinating, where the rest of the nut is the cotyledon. It's kind of the stored energy that's going to help that growth. So we take these embryos, we can actually isolate them. This is a whole cluster of embryos. Again, you'll get to see some of this in our lab. But this is the tissue that we actually transform. So we expose whole clusters of embryos to the Agrobacterium. The Agrobacterium is moving this DNA. It's moving the DNA that we've put in. And so some of these embryo cells take up the new DNA. And if those individual cells can grow into whole embryos, and then if those whole embryos can continue to develop, we take them through this process, and they start as little white blobs, and then they turn into little yellow blobs, and then they turn into green blobs, and we're heading in the right direction. And then they turn into little buds that look almost recognizable as mini plants, which are then grown into chestnut shoots. You saw some examples if you were here last night from the other tissue culture people who were talking about chestnuts. And this is kind of the stage where if you're just using tissue culture for propagation, you can start directly with the shoots. But that's not really relevant for our transformation because we need to start with these embryos. So we have processes for working directly with shoots if we want to propagate something. Hannah and Linda are experts, so hopefully you'll get a chance to talk with them if you're interested in this. But starting with the embryos is really key, and then moving them through this whole process. Once we have as many shoots as we want, we can start inducing them to grow roots, and then eventually into whole plants. So then we have a potted plant that came out of tissue culture. For many of the American chestnut lines we work with, though, these tissue culture plants are not really optimal. There's something about the connection to the roots. They can survive, many of them do fine, but they're not as robust as trees that grow from seeds. Whether or not they have new genes, just the process of coming out of tissue culture is difficult for some lines of American chestnut. So most of the time what we do is leave them in pots and induce them to make flowers. We have a rapid flowering procedure where we can grow potted trees and get catkins in about a year, and then we collect the pollen and take that outside. As soon as the pollen is used to pollinate a wild American chestnut tree, we collect the seeds, and those grow fine. So this first generation is often completely indoors until we can make a seed, and then we have collections of seeds, some of which inherit the transgene, and seedlings are what we're generally using. This also fits into our goals for genetic diversity. In contrast to agricultural cultivars that are intentionally kept consistent, and clonal cultivars are often beneficial for that consistency, for a restoration project, we want as much diversity as we can. We want to reflect the native population of American chestnut trees with extensive geographic range, adaptations to different environmental conditions, and so on. And so this, we call it outcrossing, using the pollen from this initial line and crossing it to several diverse American chestnut trees is the goal of our project. So, this sounds very technical and unnatural. Is genetic engineering unnatural? Like I said, Agrobacterium does this in nature. Sweet potatoes, in fact, exist because they were engineered by agrobacterium some thousands of years ago. So sweet potatoes have agrobacterium DNA. The more we understand about plant genomes, the more examples we see of plants with bacterial DNA, or with viral DNA, or with DNA from completely unrelated organisms. So in principle, so this paper from a few years ago says naturally transgenic plant species occur on an unexpectedly large scale. Naturally transgenic sounds like an oxymoron, but this process happens in nature. Bacteria move DNA into plants. These are unrelated organisms. DNA moves across kingdoms. So the fundamental process of genetic engineering and moving DNA between unrelated organisms is natural. For a little bit more detail about the oxalate oxidase gene, the enzyme that we've put in, this is the chemical formula, but we developed a cartoon version for a middle school presentation we did at one point where Buster Blight is using his acid attack on Charlie Chestnut. But if we give Charlie Chestnut an oxalate oxidase shield, they'll have to live in peace. And it's silly, but it illustrates the concept that both organisms are surviving, and that the oxalate oxidase is not killing or preventing the fungus from infecting the trees, but reducing the amount of damage done to the tree. So, oxalate oxidases are also found in many other plants. They're not just from wheat, so it's not a matter of some relationship to gluten or wheat allergens or something like that, because they're also found in corn and rice and sorghum and basically all of our cereal crops, as well as some unrelated plants like bananas and strawberries and several wild plants. So oxalate oxidase is natural, widely found in our diets and in the environment already. It sounds like, or you would intuitively think, that the process of genetic engineering is disruptive to a genome. And how does this, though, compare to other methods, some of those other alternatives that I talked about in terms of how many changes there are to the genome? This was studied in soybeans, where there were examples available of transgenic soybeans and traditionally bred soybeans from hybrid breeding and mutagenesis, or using radiation to induce changes. Authors found that the structural changes, so this is apart from the intentional insertions or so on, but other changes to the genome were about tenfold different. So genetic engineering actually resulted in the fewest changes, mutagenesis resulted in about 10 times more changes, and traditional breeding resulted in 10 times more changes than that. So this is just kind of a comparison of the types of, or the scale of changes in the genome. I have another illustration for this later too. So we have many options. If we want to restore American chestnut trees or do something about chestnut blight, these are some of the ones that I've talked about. Or we don't have to do anything. If we don't want to do any of these options, we don't have to. That's an option. We don't have to do anything. But my questions here are, which of these options, if you look at all of them, which have risks? Kind of a trick question. They all do. Even if we don't do anything, even if we avoid the problem and don't do anything, American chestnuts are going to continue to decline. They're not going to become healthier. Other tree species are going to continually decline and not become healthier on their own. Another question is, which ones are regulated by the government? I'm going to talk more about this. Only transgenesis, only genetic engineering is regulated. So I talked about this a little last year. This is a controlled inoculation where we intentionally infected trees with chestnut blight. The transgenic trees on the left resulted in a very small canker. The non-transgenic trees resulted in much bigger cankers. And we have good replicated data from this. This is a result from that experiment also of how the cankers grew over time. Brown is non-transgenic, and the cankers got bigger over the summer. Green is transgenic, and the cankers grew much more slowly. Again, these are controlled conditions where we're able to get good replication. Maybe more interesting, though, is what happens in nature. What's going to happen to these trees as they're naturally exposed to the chestnut blight? On the left is a darling American chestnut with oxalate oxidase. On the right is a closely related tree from the same family that didn't inherit the transgene. A little hard to see from this scale, so zooming in, the wild-type chestnut has multiple stems that are dead from chestnut blight. These are advanced cankers that have completely girdled the tree, and these central stems are dead. The resprouts are doing fine so far. In contrast, the transgenic tree has several cankers that are swollen. They have this really unique blocky appearance. The tree is definitely responding to blight and not dying from it. That said, some of our transgenic trees have died from blight. There is a range of responses that we're seeing, and the environment plays a role in this. The virility of the blight fungus plays a role in this. So there are several factors that affect the outcome of a tree disease, but in general, darling chestnuts are tolerating blight better than their non-transgenic relatives. What we're seeing is that this is the best option for American chestnuts that's currently available. So I wanted to share some updates also about what's going on with our darling chestnuts. You might have heard some news recently. One of the biggest is that the names that we were using were incorrect. So we had been calling this line Darling 58. Due to a labeling error in 2016, that was incorrect. It was actually Darling 54. These have the same background, the same American chestnut line. They have the same new DNA. It's just inserted in a different place in the genome. So we're in the process of correcting this name. This was discovered with the help, kind of concurrently, with the help of a few different collaborators. And as soon as we understood this, we reported it to the regulators, and we continued to do more testing until we were confident that that all of the trees we were working with fell into this category. So we are in the process of submitting updates to the regulatory agencies to correct this name change, and updating those documents.
Who's the regulatory agency for naming something?
There are, I have a slide for it, but there are 3 different agencies in the US that regulate these, from the name to the product to the environmental interactions. All 3 are involved. So, like I said, there are a variety of responses, even within our transgenic trees, different ways they respond to blight. In many cases, they look fine. We don't see blight infections naturally. In other cases, we see those kind of unique-looking cankers, and in a few cases, we see trees die. The important part for us is that these look better than non-transgenic trees. And in every case where we've been able to look at average canker sizes, the transgenic ones look better. They're not perfect. They're not blight-proof. This is not a silver bullet. But that might be reassuring for people who are concerned about impacts on the blight fungus, that the blight fungus is continuing to infect trees and it will not be selected to evolve and overcome this mechanism, perhaps. Some of the darling trees grow slower than their non-transgenic relatives, and I have some examples of that in the next slide. The insert location is different, and another concern that was initially brought up kind of at the same time as a name change is that trees, some people initially assumed that trees couldn't inherit 2 copies of the transgene. And so the darling trees that we created have 1 copy of the transgene on 1/2 of the chromosome. And so that means that as those chromosomes split and make pollen cells, if you remember the basic biology, that about half of the offspring of these trees get a copy of the transgene, the other half are non-transgenic trees. And that's basically what we would envision would be the scenario in a restoration context. But after several generations, you could take 2 transgenic trees that each have 1 copy of the transgene and cross them together and potentially accumulate 2 copies of the transgene. this is what we would call a homozygous state, if it has 2 copies of the transgene. And there was a concern that for some reason that couldn't happen, or that homozygous trees weren't healthy, or that homozygous trees weren't developing, something like that. That's not at all what we've seen. We have homozygous trees that look great. We're propagating them, we're growing them in our growth chamber, and in fact we have pollen from homozygous trees that will let us enhance our breeding program and hopefully get even more transgenic offspring. All right, I think my next slide was about the height growth of the growth rates of the darling trees. And on average, if we look at all of the darling trees compared to their closely related siblings and trees from the same cross that don't have the transgene, the darling trees are a little shorter. They seem to grow a little slower. We are trying to understand this and figure it out. One of the things that we're seeing, though, is that this doesn't apply to all of the families. As we're, like I said, outcrossing the pollen onto diverse American chestnut backgrounds, some of them show more of a growth rate difference than others. Some of them look about the same as their relatives. And so we're, at this point, focusing on those families that have the best characteristics, that have the best growth rates, that have the best form. We're collecting pollen from those best trees and using that, prioritizing that in subsequent generations of the breeding program. Before I move on to that, I will get back to that. I want to mention one other point under our current status about non-exclusive licensing and distribution strategies. We think it's really important that there isn't just a single group, a single entity who's entirely in charge of these trees. So our university is working on licensing agreements where different groups can become involved in this and can help distribute trees at different scales for different purposes. We want to be sure that these trees are available and usable for restoration, for research, for education, and we're starting to think along those lines. Difference between Darling 54 and Darling 58. I said it's the same genetic background. So this dark blue represents the background. They're the same. The insert location is the little blip that you can barely see. I tried hard to make this to scale of the genome. And so it is really difficult to visualize the difference. In contrast, backcross trees have some 6 to 10% or more of their genome from Chinese chestnut trees. And so there are many more sites across a backcross tree or a hybrid where the DNA was interrupted, where the insert location of a gene, if it causes disruptions, there are many more of these in hybrid trees than there are in our transgenic trees. And this is not to say that backcross trees or hybrids are scary. Like, this is not dangerous. The trees are not harming people or the environment. But I would suggest that the same criteria be applied if we're concerned about insertion sites for these trees. So I'm going to move very quickly through, these are some examples of height growth. My point was that height was not that different. On average, the darling trees are a little shorter, but there are some families where they are really similar. I talk about several types of environmental interactions. I'm sorry to skim through this and move quickly, but please find us and we can talk more about this. Several types of environmental changes. And I would suggest that if there are concerns about environmental changes, that those should be considered for other types of restoration trees too. We have not seen differences in mycorrhizal relationships or use of pollen by bees. You're going to hear about some physiology, some respiration measurements. One of the ways that we're looking at these differences in growth rates is in respiration, how the trees are using carbon dioxide and oxygen, and whether the production of this enzyme is resulting in the tree expending more energy. And we're really not seeing major differences there. So this is Taylor. She's going to be giving one of our field tour stops, so you'll hear more about this on Wednesday. I talk about the regulatory process. 3 different agencies are all involved. We are currently in the process of submitting revisions with these name changes. And then after that is concluded, we expect that future submissions will go much smoother. So part of our rationale for continuing to pursue this, for correcting the name, for submitting these changes, is that this will be a really important basis for future submissions as we can look at some next steps and improvements and iterations past Darling. This first submission will really lay the groundwork, will really be an important step for those future submissions. We're intentionally focusing on collaborations with educational partners, with continued research, and very quickly, an important point here is that large-scale forest restoration is not going to happen suddenly. This is going to require a lot of work. It's going to take a lot of time. Even after the regulatory approval process, we're talking about decades of scaling up production and continuing to do studies, continuing to collect data before this happens. It's going to be intentional, relying on input from people. I unfortunately need to move quickly. I want to mention that we are looking at some other trees. So these are nematodes that cause beech leaf disease, a brand new, 12 years ago it was introduced to the US. Disease on beech trees. We're just starting to look at beech trees. We don't have transgenic beech trees, but we're trying to understand this better and think about whether the similar procedures could be applied toward other types of trees. Likewise, emerald ash borer is causing a lot of damage. We don't have transgenic ash trees, but we hope that some similar technologies like we've started to use on chestnut might be able to protect other trees as well. So with that, sorry to rush through, but thank you for your time and attention, and please find us with more questions.
Okay, thanks, Andy, for that short presentation on a very long topic.